Hello, everyone, and welcome to the Foundation Fighting Blindness National Chapter webinar. We will get started here in just a few moments. Hello, everyone. As you're joining, we are getting ready to start here right at the top of the hour. Hello, everyone, and welcome to the Foundation Fighting Blindness National Chapter webinar. My name is Bailey Bostelman, Director of Chapter Engagement for the East. We're so excited to be with you today, and thank you for sharing your Saturday with us. I would briefly like to review a few logistical details for the call. Currently, all participant lines are muted. Today's call is being recorded and will be shared with all registrants after the meeting concludes. You will also be able to find the recording on the Foundation Fighting Blindness website within the upcoming days. If you are using a screen reader, please be aware that the controls are at the bottom of the Zoom interface. Control bar may collapse when not in use, so make sure to joggle your mouse before clicking anything. If you prefer to prevent the controls from auto hiding, go to your settings within the Zoom platform, select Accessibility, and then select Always Show Meeting Controls. If you have any questions throughout the presentation, please send them using the Q&A function or send it to chapters@fightingblindness.org. Foundation Fighting Blindness Chapter Network brings together those across the country to join in fighting blinding retinal diseases. Our family of volunteers and leaders stand up for those impacted by blinding diseases. From day one, they share the latest research and advancements and provide access to local resources, guide individuals through their personal journey. And together, they celebrate the Foundation's mission. To get involved in one of our 45 chapters across the nation, reach out to us at chapters@fightingblindness.org. Now I'm going to hand it over to my counterpart in the West, Nathan Klucke, to share a little bit about our Raising Our Sights program.
Thank you, Bailey. So Raising Our Sights is an opportunity to combine your commitment to the Foundation Fighting Blindness and bring your fundraising vision to life. So with your creativity, passions, and connections, you can create a small personal fundraiser or a large community event, all to help raise awareness and funds to find treatments and cures for blinding diseases. Raising Our Sights is a perfect opportunity to get your entire community involved while contributing to our mission. And that is exactly what our Colorado chapter president, Lindsay Blankenship, did when she created her incredibly unique and ingenious Raising Our Sights event called Wine for a Cure. With the help of one of the best wineries in Oregon, the Stoller Family Estates, Lindsay has created 2 featured labels that are raising awareness for the foundation, and 30% of the proceeds from each bottle go directly towards our mission. So whether it's a custom wine, a bake sale, running a 5K, hosting a pub crawl, or even a push-up challenge amongst friends, we encourage you to start your own unique fundraiser through Raising Our Sights program. And you can learn more at fightingblindness.org/raisingOurSights. And now I'd like to welcome our VP of Science Communications, Ben Shaberman.
Well, thanks, Nathan, and thanks, Bailey, for kicking us off for this webinar on stem cell therapies. It's great to have all of you out there with us. A happy Father's Day weekend. Tomorrow happens to be Paul McCartney's birthday for all you Beatle and Paul fans. But we're very excited to have a great lineup of cell therapy researchers with us today to present what they're working on. We have Kapil Bharti from the National Eye Institute, Karl Wahlin from the University of California, San Diego, and Henry Klassen, who's with jCyte and University of California, Irvine. So they're each going to present for somewhere in the neighborhood of 12 to 15 minutes. Then we'll have some time for Q&A from you. You can enter your questions in the Q&A section, as you heard about. Remember, these are cell therapy researchers, so questions around cell therapies are appreciated. And then to close things out, our esteemed Chief Scientific Officer, Claire Gelfman, will deliver the closing remarks. So before we get started with our expert presenters, I just wanted to provide a brief overview of stem cells and potential applications for cell-based therapies. And there are really 3 or 4 ways that we are using cell-based therapies and stem cells in the inherited retinal disease and dry and wet AMD therapy space. One way that cells are used or are being explored is for replacing lost retinal cells. So as I think many of us know, the cells in the retina that are so important for vision are photoreceptors. There's another layer of cells called RPE cells, which provide support for photoreceptors. Those are really important too. And often in retinal diseases, these are the cells that are affected. And so the idea with a cell-based therapy could be to replace those lost cells for restoring vision and even just saving vision. Now, another approach for cell-based therapies is to inject cells so they release growth factors and proteins and signals to save someone's existing photoreceptors, to preserve their vision. We often call this neuroprotection. And you can think of these cells as being like a drug factory to help keep a person's existing cells alive and preserve vision, or at least slow vision loss. And then finally, we can also use cells, stem cells, to make mini retinas or retinal organoids to study retinal diseases in a dish. If we're using human retinal organoids, we can better understand how disease impacts these human cells. We can also use these organoids or these mini retinas to screen drugs and other therapies. And again, because they're often human cells, it's a nice way to see if these therapies may work in humans. Now, just about everything I've talked about is usually derived from stem cells. And let me give you a quick definition of what a stem cell is. The really simplest way to put it, kind of the fun way to put it, is a stem cell is a cell that doesn't know what it wants to be when it grows up. It's a clean slate. It's very primitive. And what we call a pluripotent stem cell is a cell that can become just about any type of cell in the body. And of course, in our world, we like to make photoreceptors and RPE cells. And so we're taking pluripotent stem cells and making them into these cell types. Now, another aspect of stem cells that's very beneficial is from a small sample of stem cells, you can literally make billions and billions of the cells you need for therapies. So they're very easy to replicate. And therein lies the real power of stem cells. You can make them into just about anything, and you can readily replicate them. And that's, again, what we mean by pluripotency. Now, another thing I think is important for us to understand is we usually don't put stem cells in their primitive state into the eye or really any part of the body because you want the cell to be mature so you know what it is or you know what it's about to become. If you put a raw stem cell in the body, you have less control over what it could become. And it could become something that you don't want, like a tumor cell. So while we often talk about stem cell therapies, the actual therapies are cells that are derived from stem cells. So thanks for letting me make that quick clarification. And another aspect of cell-based therapies that can be very beneficial is in many cases, they're gene agnostic therapies, meaning they're designed to work regardless of the mutated gene affecting the patient. So they have the potential to help a lot of people because they're not gene mutation specific. And today, with the three presenters you're going to hear about three very different ways that these cell-based therapies are being used in retinal disease. So with that, I am honored to introduce our first presenter, Kapil. Kapil, you can begin sharing your slides while I introduce you. Kapil Bharti holds a PhD, and he joined the National Eye Institute in 2012 to lead their ocular, stem cell, and translational research section, which conducts research on degenerative eye diseases using induced pluripotent stem cell technology. And I'm sure you're going to hear more about what that is. Under Bharti's direction, the lab has developed in vitro disease models and high throughput drug screens. And most notably, he's marshaled preclinical work leading to the first clinical trial in the US to use replacement cells and tissues from patient-derived induced pluripotent stem cells. And that's what you're going to hear about. And excitingly for Kapil and the NEI, he was named scientific director of their entire intramural research program recently. That's all the internally led research. So congratulations, Kapil. From there, Kapil, I'll hand the talk over to you.
Thank you, Ben. Can you hear me fine?
We can hear you fine, and
wonderful.
Slides are up. Thank you.
Wonderful. Thanks for the kind introduction. First of all, a big kudos to the foundation for organizing this cell-based therapy awareness chapter. And thanks for having me here. And Ben, again, thanks for doing the overview. I'm going to build upon this to kind of further go into details of the work that we are doing. So this works. All right. I think I don't need to tell this audience that vision loss affects every aspect of life. There are over 350 different genetic mutations that lead to various kinds of vision loss, ranging from night blindness to peripheral vision loss, encroaching towards the central vision, and diseases that start with the center and spread towards the outside. These diseases range from retinitis pigmentosa to Best disease, macular degeneration, Bardet-Biedl syndrome. These are really devastating conditions, and for most of these conditions, currently we do not have any treatment. If you look at the back of the eye, it's very clear that most of these 350 genes or mutations and conditions affect primarily these three tissues in the back of the eye: the photosensitive cells, the photoreceptors, the retinal pigment epithelium that is sandwiched between the photoreceptors, and the blood supply that is present in the back of the eye. This tissue, RPE, is a highly polarized tissue, and throughout its life, it is maintaining the health and integrity of the photoreceptors and also of the choroidal blood supply. At the same time, this tissue is maintaining the outer blood-retinal barrier and allowing the nutrients from the blood supply to go to photoreceptors and the metabolites from the photoreceptors back into the blood supply. So naturally, when any of these genetic mutations affects directly the photosensitive cells or the support structure or the blood supply, all the three cell types suffers, and that results in vision loss, and in extreme cases, that results in blindness. Here is another example of three tissues at a higher magnification, and just so you can see and appreciate the close proximity of the three tissues of photoreceptors, the retinal pigment epithelium, the RPE, and the choroidal blood supply. And as I said, different mutations can affect different aspects of these different tissues. There are mutations that primarily affect the RPE, which will result in loss of photoreceptors because they support the photoreceptors. They will also result in loss of the choroidal blood supply. There are gene mutations that directly affect the photoreceptors, and they die they would die because of the mutations or start dysfunctioning. But at the same time, that mutation or that defect of photoreceptors can lead and spread to death and dysfunction of the RPE and the capillaries. Hence, the symptoms start to spread across the entire eye. So really, depending on what the kind of disease is, depending on what the stage is and how many different cell types are affected, we have to come up with different types of treatments. So now as a schematic, just to orient you, we're talking about neurons, interneurons, the three cell types that I talked about, photoreceptors, the RPE, and the blood supply would be underneath. So if we go from the healthy state, which is here, you can try to stop disease by prevention, which happens in very small number of cases. As the disease progresses and one cell type, for instance, in this case, photoreceptors are affected, that's called disruption of that cell type. It can be and has been in some cases to some extent successfully been repaired or corrected by gene augmentation or gene editing. But the prime, as Ben pointed out, that the prime way gene augmentation or gene editing would work is you have to have that cell type still there so you can augment the missing gene and or edit the mutated gene and then repair the function of that cell type before it dies off. But if in cases where the cell type is missing, for instance, now you go to this case where photoreceptors are missing or the RPE is missing, the only way you can repair is by providing a cell replacement therapy, the cell-based therapy that we're going to talk about today. In this case, an RPE patch or photoreceptor. Now, further, more extreme cases where the tissue is really degenerated, the whole retina structure is disarrayed, that's when we talk about multiple tissue replacement. It's called a tissue disarray. We talk about tissue replacement therapy. I'll touch upon this towards the end of my talk. That is work in progress in our lab and many other labs, where we're now trying to combine stem cell derived tissues in a dish, multiple tissues for receptors, RP and the choroid, with the goal to bring them all back to patients. So Ben mentioned this, to develop a cell-based therapy, we have to start with a stem cell. A stem cell, by definition, is a cell that can replicate itself. So that's a classical definition of a stem cell, that it can replicate and make more copies of itself, and at the same time, it can make other cell types of the body. And here's an example of multiple cell types listed here: neuron, fat cell, blood cell, muscle cell, eye cells. So a stem cell has a potential to differentiate into any cell type of the body. There are primarily three types of stem cells: embryonic stem cells, which are pluripotent in nature. That means they can make any cell type of the body. These stem cells are derived from a pre-implantation blastocyst from an embryo. The second cell type that Ben pointed towards is induced pluripotent stem cells. These are also pluripotent and for all practical purposes are identical to embryonic stem cells. But the beauty is that we can make these cells, make the iPS cells from any adult. We can take a snippet of your skin, we can take a few drops of your blood and reprogram that into iPS cells. It takes only a few weeks to make iPS cells from your own somatic cells, from your own body cells. And these cells are going to work just like ES cells and will make any adult cell type of the body. And then the last cell type that I guess Henry will talk about in his talk is tissue-specific stem cells. These are unique stem cells that can make specific, these are multipotent stem cells that can only make specific tissues in specific cell types in specific tissues. So for the purpose of my talk, I'll focus on iPS cells. And I'll talk about that, how iPS cells are first made and how we can then differentiate them into any other tissue. So like I mentioned, we can start with a snippet of your skin. We really need 2 millimeters of your skin and grow the fibroblasts from your skin in a dish, or a few drops of your blood, expand them in a dish. We add what we call reprogramming factors. These are proteins, just a few handful of proteins, 4 proteins actually to be specific, that are added to these cells. And we just wait for about 3 to 4 weeks. And in this time, these proteins have the power to convert these skin cells or blood cells into iPS cells, into these pluripotent stem cells. And then from these, we add specific growth factors to then make them into any other cell type of the body. In our case, what we have developed is a replacement cell type to make the RPE cell as a replacement cell type for a disease called age-related macular degeneration, AMD, where RPE cells degenerate, and as a result, photoreceptors die and choroid die. So like I mentioned, we start with blood cells, we convert this blood into iPS cells, and then we add the right growth factors, right chemicals at the right time point. This process takes us about another 10 weeks. So 3 months from blood cell to iPS cells, and from iPS cell to RP cell, fully mature RP cell in about 10 weeks. And this process, as you can see, the schematic shows how the shape of cell is changing. And it's critical because not only the shape of cell is changing, they're acquiring the functions of native RP cells. These apical processes, these hair-like projections that you see on top of RP cells, this is how they interact with the photoreceptors. These structures are quintessential for the function of RP cell. So now we know that when we make this from patient cells in a dish, these cells are not only structurally similar to the cells present in their eye, but they're also going to function like cells present in their eye. So when we transplant them, they're going to behave like native RPE cells. So we develop a patch. So we put in purple here is a patch that we develop here. These purple cells are RPE cells. These are made in vitro. We put them on a scaffold and we develop a procedure, which you can see on the top right here, with a cannula. The patch goes inside the cannula, and the cannula has a shape that really fits the curvature of the back of the eye, and then through which we can actually deliver the patch right under the retina, right where photoreceptors are degenerating. And the goal is that this patch would really situate under the degenerating photoreceptors, which are missing the support from the RP, from the RPE that has already degenerated, and will replace that degenerated RPE with a new, fresh RPE, young, healthy RPE, and eventually stop the photoreceptors from degenerating. And actually, surgically, this kind of approaches have been tried. Surgeons have in the past cut out a piece of RP from the same eye and moved into the periphery. And this procedure has worked. Now we have modified this to make it from patients' iPS cells, made it more high throughput, made it more high content, and we can also quality control the cells much better when we make them in a dish. So this work, just to take it to patients, I want to emphasize how the process of taking the cells to patient works. So we start with proof of concept data in the lab with working with animals where we make iPS cells, make the RPE, and we test their safety and efficacy in animal models. This process itself can take anywhere between 5 to 8 years. And then we take them to patients. Right now, our work is what we call a phase 1 study, where we're testing the safety of these cells in a single center study at the NIH, at the National Eye Institute, where we're testing the RPE patch made from AMD patients on stem cells in a phase 1 safety study. So this process from phase 1 to phase 2 can take anywhere between 2 to 5 years. And then from there, we expand it to multi-center trial. And at that point, we are also measuring, we have enough patients that we can measure statistically different the efficacy of the patch. And at that point, we get approval from the FDA to use it as a common medical practice. So just as I pointed out, the work that we are doing currently is in phase 1, and we hope to continue that into phase 2. I want to briefly here emphasize that we have to go beyond treating, just replacing just one layer. Here, I'm showing you the idea that now we are actually making all the different cell types of the back of the eye from patients on iPS cells. We again start with patient somatic tissue, make iPS cells. I showed you that we can make the RPE, but now we can also make the choroidal blood vessels. So you can see an example of this video where we bioprint the cells of the choroid to make these vessels. And then on the left side, you see an example of differentiated photoreceptor progenitor cells made from the same iPS cells. So now we put all 3 cell types together to reconstruct the entire back of the eye and to be able to replace the entire tissue in case of extreme stages of late stages of degeneration. At the same time, we are, and others are working on techniques to genetically edit the mutation that was present in the patient's cells. So then when we deliver the tissue back to patient, it's healthy tissue, not diseased tissue anymore. So with that, I want to actually end with 2 slides where I want to caution patients about difference between approved and unapproved stem cell therapies. You will hear a lot about stem cell clinics, people who are offering stem cell injections for a certain amount of money. Most of these are often unapproved and unproven stem cell therapies. Will caution patients that in many cases, patients have been hurt, the vision has been actually lost by use of those injections of these so-called stem cell therapies. Most of these are not FDA approved. They have not been tested, rigorously tested through the IND enabling process that I just showed you 2 slides ago. So please be careful. Any authentic stem cell therapy will never ask you for money when you participate in a phase 1 trial. If patients are interested in more information around that, there are 3 places to look for it: at the FDA, the NIH, and International Society for Stem Cell Research. They all provide information and a difference between stem cell therapy and stem cell derived therapies. So with that, I'll stop there and hand it back to Ben. Thank you very much for your attention.
Thanks for that awesome talk, Kapil. And I'm really glad you included that last slide on bogus stem cell therapies. One key aspect of those is usually you have to pay for them. And you shouldn't be paying for a stem cell therapy that's in a trial. So that's a red flag. And feel free to reach out to us at the Foundation Fighting Blindness, info@fightingblindness.org, and we can help you understand if a therapy is legit or not. And Kapil, I really think it's so cool that you're using patient cells to not only make RPE, but photoreceptors and choroid. It's really great work, and we wish you the best on your clinical trial as it progresses. Thank you.
Thank you, Ben.
And again, if you have questions for Kapil, you can start entering them in the Q&A section. And once we're through all the presenters, we'll get to as many of those questions as we can. So next up, it is my pleasure to introduce Dr. Karl Wahlin. Karl holds a PhD. He is an assistant professor of ophthalmology and the director of the Richard C. Atkinson Laboratory for Regenerative Ophthalmology at the University of California, San Diego. His lab leverages stem cells and genetic engineering to uncover the basic biology of eye development. And by gaining a better understanding of how the eye develops, they can develop new tools to combat debilitating eye diseases like retinitis pigmentosa, AMD, and even glaucoma. So, Dr. Wahlin, your slides are up. Take it away.
Great. Thanks for the intro, Ben. And, it's a pleasure to be here today. It's really great. So welcome, everyone. So unlike the other speakers today who are going to be describing very impressive research programs that are already in the clinical space, my work is actually at a much earlier phase, but I hope over time it's going to create an equally exciting alternative that'll be broadly applicable to retinal degeneration stemming from lots of different causes. And so we know there's close to 300 genes and loci, which when mutated cause disease. This is a problem logistically in terms of generating gene therapies and all of the other approaches. So what I'm going to talk about today.
We're getting live.
He's live right now.
Mhm.
Where is he?
At his office.
Is there a problem?
Can we make sure everybody is muted except for Dr. Wahlin? Thank you.
What?
Okay. Okay, great. So for many years, researchers have really worked extensively on ways to reduce and slow down progression of retinal degenerative diseases. This includes application of neurotrophic factors to preserve cells, gene therapies to compensate for lost function, and transplantation to replace lost RPE or provide trophic support to remaining photoreceptors. So the first and the third talks today pretty much cover those sections. These are areas that show a lot of promise, but when photoreceptors have already died, we need another approach. What I want to talk about today takes a cue from the animal kingdom, and this is an area called endogenous regeneration. So what is this endogenous regeneration? Well, there's many species, including amphibians, birds, fish, that can replace neurons lost to injury. And this occurs from existing support cells called glia, which are activated in such a way that these cells can multiply. Some of these become new neurons, and some of them replace the support cells that were there to begin with. Now, this sounds a lot like science fiction, but tissue repair happens all the time in nature. Lizards grow new tails, amphibians grow new limbs, and some species like flatworms, well, they can grow an entirely new head. That's pretty cool. So, many species, actually, including fish and amphibians, can also grow retinal cells relatively easily. And so this is sort of important for us. Mammals like us, on the other hand, we don't seem to be capable of replacing these neurons quite so easily. But we think there might be a biological template that's been silenced through evolution, and under the right conditions, we think that we might be able to reactivate these repair mechanisms to fix the human eye as well. So there's a lot of evidence that these so-called silenced biological instructions can be reactivated. An example that always comes to mind for me is that chickens don't normally grow teeth, right? That's a pretty horrifying thought. But actually, scientists have found chickens that grow new teeth. And these teeth are very alligator teeth-like, even though birds have lost this capability over millions of years. And since the time that they've identified these chickens, they figured out some of the mechanisms that happen there. And so in a nutshell, they were able to identify the mechanism and prove once and for all that these sort of silence programs exist. So we'd like to think that these same sort of things happen in the human eye as well. So we know that many species, including fish and amphibians, can regenerate new neurons. Very cool. Recent studies also shown that some mice can also make limited numbers of neurons after injury. And so one of the main challenges here is getting the right types of neurons to form. If photoreceptors have died, we want to replace photoreceptors. If ganglion cells have died, we want to replace those. And we want to do them in sufficient numbers that you can restore vision. So why are we so excited about endogenous regeneration? Well, as I mentioned, there's hundreds of genes and loci which, when mutated, cause disease. And it's going to be a very hard challenge to find a gene therapy for all of these. But with endogenous regeneration, you could potentially flip that switch, restore new neurons. And even if the cells still have the underlying mutation, you might be able to give them enough time that the neuroprotective strategies can do their job. And also this might lead the way for a merging between gene editing to repair the mutations as well as restoring. So again, these kind of concepts are applicable after the cells have died, because after neurons have died, they're not coming back. So my lab's been studying different ways to reprogram human cells and Mueller glia into new neurons with the hope that we can restore vision by replacing those lost neurons. So generally, our lab focuses a lot on a disease called Leber congenital amaurosis. This is an early onset form of retinitis pigmentosa. There's about 20 genes which, when mutated, cause this disease. This affects primarily the photoreceptors and RPE complexes, so the cone photoreceptor on the left, the rod photoreceptor right, and the RPE. And there's many different genes, as I mentioned, that affect different parts of the cell and different cellular functions. And if any of these are dysfunctional, the cells will die. This is a relatively rare disease, 2 to 3 per 100,000 newborn cases, but the underlying mechanisms could be similar to many other forms of retinal degeneration. So the regeneration that we're talking about here is not just restricted to this disease, but also many others. So in our lab, we leverage human pluripotent stem cells quite heavily. Typically, when we make them in the lab, we use isolated white blood cells, which you can see here. We use specialized tubes and collect the white blood cells. This is what they look like when you look at them under the microscope. We take these white blood cells, we introduce a virus that delivers 4 proteins. The proteins are not important, but suffice it to say that after a couple weeks, these floating white blood cells will convert directly into stem cells. From there, you have an unlimited supply of cells, which Ben has already described, which you can then use to form any types of tissues in the body. These basically live forever if you take care of them correctly. We then gene edit these cells. So by gene editing, we introduce fluorescent reporters into these cells and then differentiate them. And this differentiation can take up to 8 months to a year. So it takes a very long time. Remember, a human embryo takes 9 months to form. Well, these human stem cell-based systems, they typically take on that sort of order timescale to mature and differentiate. So it's very important that we have ways to visualize the cells and track them over time. And that's what our lab focuses a lot on. So different things you could do with stem cells. You can study them to, you can use them to study disease. Our lab uses organoids, but some other labs might study flat cells growing in a monolayer. We can use these cells to block disease. So we can screen for neuroprotective drugs. You have a scalable system where you can make millions and billions of cells and screen for drugs or test different gene therapies. Or you can use these to replace lost cells. You can make new RPE cells, photoreceptors, ganglion cells. So depending on the disease you're looking at, you can make new cells. Often we talk about transplantation for this, but there's another angle, which is endogenous regeneration, where you can simply introduce a virus or some other small molecule, something to trigger that conversion inside the eye. So you wouldn't be transplanting, you would be making the new cells in the actual place that they actually reside. The way we make organoids in the lab, we use these 96-well round bottom plates. We dissociate stem cells. We put a defined number of them into each of these little wells, and gravity brings them down. They spontaneously start forming neural tissues if given the right recipe. So down below, you see a typical recipe that we use to make an eye. We use small molecule chemicals to block some pathways, other small molecule chemicals to activate pathways. The pathways themselves are not important, but we do have a well-defined cocktail of chemicals that we add to the cells in these little plates, and then they start spontaneously forming brain tissues and retinas, and then we isolate them. So I mentioned it takes a very long time to grow these, and without the ability to know what they are, this could be problematic. So we use gene editing. So CRISPR-Cas9 gene editing is something which has already, despite its only 10 years being used in the lab, it's already achieved Nobel Prize status. So this is something that is such a powerful research tool. It allows us to create mutations in cells so we can study disease, we can repair mutations, we can deliver genetic cargo. And in our case, we're talking about inserting fluorescent tags so we can visualize cells. And so we've used this kind of approach to visualize rod and cone photoreceptors. This is an example of an organoid, a retinal organoid that was grown for 173 days. And you can see these different colors here, which represent different colored fluorescent reporters which were introduced to the cell. So anytime these cells are produced, the fluorescent reporter lights up and we can track them. And this is very useful if we're studying disease modeling. A lot of our effort, again, is Leber congenital amaurosis. So if we can track these cells over time, we can watch them form, but we can also watch them die, and we can watch them be preserved after we've done a certain treatment. So very, very interesting tools that we have. Another research area that we're really interested in is developing genetic switches to make different types of cells. So, I mentioned earlier that it's not sufficient just to make progenitor cells. When we're doing this endogenous regeneration, we need to get the support cells to divide, but we also need them to turn into the right types of cells. So leveraging our basic developmental biology, we can take genes that we think are involved in a cell process. We can introduce them into genetic switches, turn these genes on, and then we can watch what happens with these cells. So in a matter of a week, we're able to take stem cells, flip our switches, and make greater than 95% positive retinal ganglion cells in a dish. And this is a huge feat for us because now it tells us that we know the genes that are important if we want to make new retinal ganglion cells in the eye. It also gives us a very scalable way to grow these cells if we want to drug screen. So neuroprotection is also important. So we can grow millions, potentially billions of these cells. We grow them on a plate. We can screen for FDA-approved drugs, and we can repurpose ones that have been used for other purposes and see how they work in retinal cells. And so this cell engineering approach has allowed us to make new cells and then confirm that certain drugs are neuroprotective for retinal ganglion cells. So we're here to talk about photoreceptors, though. So the good news is we've been able to accomplish a similar type of goal with cone photoreceptors. The efficiency is not quite as high as the retinal ganglion cell work that I just showed, but it is very high enough that we can improve and optimize the system. And it's given us some really nice clues as to the genes that are most important for cone cell development. So we're going to use this approach in the regeneration strategy in order to take proliferating progenitor cells and push them to become cones. So, we've given a little bit of lip service to the fact that other species can create new cells and regenerate from the inside out. This is an example of a zebrafish that was injured after 3 days. This is from the Perkins lab. What they showed is that 3 days after injury, most of the photoreceptors have died. 30 days later, most of them have come back. So this is remarkable. So this goes back to that underlying program that we really hope is there in the humans. And so part of the cell process is that after injury, we need proliferation. We need cells to become new neurons, and we need them to become the right types of neurons. In zebrafish, this works, seems to be limited in humans currently. Now, the proliferation thing is a concept that other labs have been focusing a lot of effort on. In fact, one group from the Blackshaw lab has done an extensive analysis comparing what happens after injury in multiple species. And they looked at chickens, and they looked at zebrafish, and they looked at mice, and they found some pathways which were different between the mice. So this gives us targets that we could potentially apply to the human system and push them in the right direction. Mice, there is limited regeneration potential in mice. A recent study this past year did in fact show that some new retinal ganglion cells could be generated. I think the efficiency probably is something that could be higher, but this is a really reassuring and very exciting finding. So how are we going to do this in humans? Well, we're using our human retinal organoid model up in the top left here with different reporters. We're using information that we've learned from the induced neuron approach, this cell conversion strategy with genetic switches. Now we know which genes are probably important for making cones. We have a system where we can grow the retinas. So this is all of the layers of this is the important layers of the retina we're talking about. We can kill photoreceptors using different strategies, light, toxins, mutations. And then we would use a viral-based strategy to trigger proliferation of these support cells. So this support cell here, Mueller glia, becomes many different proliferative cells. Some of these get instructions and become photoreceptors. And in the end, we hope that this will restore function. And this is really going to rely on a viral-based strategy to deliver genes which trigger proliferation, and a second set of genes which will trigger the regenerative capacity of a progenitor into a cone photoreceptor. And so that's essentially the work that our lab is focusing on right now. So, thank you.
Great talk, Dr. Wahlin. It's very exciting to think about the possibility that a retina can regrow its own new rods and cones. I realize we're a ways away from that, but we really appreciate the work you're doing to try to figure out how to do that. So on to our final presenter. It's my pleasure to introduce Dr. Henry Klassen. Dr. Klassen has an MD and a PhD. He is a professor of ophthalmology at the University of California, Irvine, and he has a longstanding interest in neural plasticity and regeneration that dates back to his undergraduate days at UC Berkeley and continued during his MD and PhD studies at the University of Pittsburgh. He's co-founder and chairman at jCyte, which is developing a cell-based therapy for preserving vision in people with RP and potentially related conditions. And we're very excited to get the latest, an update on this emerging therapy that's been in a clinical trial. So Henry, you can share your slides if you'd like, and we'll get going.
Okay, thanks, Ben. Do you see them?
We do not, or I don't.
Because, okay, let me go back a little here.
I think you should have a share.
I think I shared it on my own, but not on yours. Okay, here we go. Share. Boom.
There you go.
Okay, and then is there one more step?
If you go to slideshow, you
Yep.
should start.
How's that?
Looking good.
Okay, great. Thanks for the assistance, and thanks everybody for the talks that preceded mine. You've done a lot of the yeoman's work here in introducing these topics and even diving well into the story. I don't really have to go over this slide except to say that we've been introduced to a wide range of ways in which cells might be used to treat retinitis pigmentosa and related disorders. And in this case, we're going to focus more on the neuroprotection type strategy. Try to keep an open mind as to what that really represents in practice. We know what the idea behind it is preserving what's there, but it may be a fascinating topic all its own. Now, in terms of the cell we're using, you've heard about the pretty much all these cells start as pluripotent cells, but that in terms of using them for an actual treatment, you're going to want something that's more restricted in terms of what it's capable of, with the idea that you want to know what it's going to do when you put it in the body. And that's truly the case here. The cell that we use, we call it a retinal progenitor cell. And the progenitor is your tip off that this isn't exactly a true stem cell. It's not pluripotent. It's now a multipotent cell, so it retains a lot of flexibility, but it's restricted in terms of making retinal cell types, hence a retinal progenitor. These are derived from allogeneic tissue, which means it's donor tissue. And we deliver these, in our case, with an intravitreal injection. So that means they're injected directly into the clear gel at the back of the eye and in front of the retina. And the image that I present here is an image in a rat eye of the human product that's been grown in a dish and then injected into the vitreous. The cells that are injected, they glom together and form these little nuggets or spheres, if you will, of progenitors. And so they can survive in this situation of just living as little clusters in the vitreous. And from there, without integrating into the host retina, they're still capable of both surviving and secreting neurotrophic factors. And these are the factors that are going to have the actual therapeutic effect in terms of neuroprotection on the host retina. So we're trying to treat the rods and cones by using these progenitor cells. And the way we're doing it in this case is to promote the survival of the host neurons. But in fact, you could use these cells in other ways. For instance, you could use them for cell replacement, but that's not the strategy here. As Ben alluded to, these cells can be agnostic to genotype. So you're not fixing the underlying mutation, but you're trying to maintain function in the diseased retina. One of the real benefits of using this approach is that the cells can be re-injected. So if the treatment effect fades off, as we expect it will, when the cells die, we can re-inject additional cells. So that brings it a little bit closer to the types of treatments we're more used to in terms of being able to redose. And the beauty of this is that these cells are well tolerated by the body without the need to tissue type or immunosuppress the patient. And that's really important because it makes the treatment a lot easier, simpler, and easier on the patient. And this tolerance extends to retreatment. So that's an important aspect of this. And now I want to turn our attention to our most recent trial, the phase 2B trial. In particular, I'm going to introduce both the trial and the results, but then focus more on the subsequent post hoc analysis. And so this trial is again, we admitted people with RP and we did not select in terms of genotype, with the idea that genotype may not matter in this for this particular type of treatment. We took a broad range of visual acuities So we were trying to be unrestrictive that way. The reason there's any restriction at all, on the low side, we needed some room for improvement because we're going to use visual acuity as our endpoint. So if we took people with perfect acuity, there's no improvement that's possible. And so we needed people had room for improvement. By the same token, we took at the higher end 20/80 because we didn't want people who had lost the ability to be measured in terms of their visual acuity, because we need quantitative measurements to get a result out the other end. And the final set of criteria related really to other diseases that might make the interpretation of the results complicated, and in particular things like macular edema. We had to rule that out, or optic nerve disorders, things that might also be affected by the treatment that would confuse the story. From there, patients who were selected were randomized into 3 groups. One was 6 million. There was also a 3 million dose. That was the highest dose from the previous trial. And then there was a sham control. And again, our primary endpoint was best corrected visual acuity at 12 months. And this was a particularly large study for RP. So it was designed to give us a lot of data to help us go into the pivotal trial. And I think we've done that, or I'll show you the data that we got from this. So looking just at best corrected visual acuity, and there were other secondary outcome measures, but I'm not going to get into that. The dark blue is the output from our higher dose, and we see what looks like a nice dose response curve here from the treatment. And it separates well from the lower dose, which is the 3 million dose, seen with a dotted light blue line down below. And then that looks fine, but then the challenge comes when we look at the sham group. The sham group had an apparent sham effect of around 3 letters, and that was high enough to cut into our statistical significance. So when we look at our final endpoint at 12 months with a higher dose, we had what looked like 7.5 letters of improvement from the treatment, but that was cut into by the 3 letters of sham effect. And so it didn't quite reach statistical significance. We had a p-value of 0.099 as opposed to the 0.05 we were looking for. Interestingly, you see there's virtual plateau from 3 months out in terms of change in acuity. And at around 6 months, the P was slightly better at 0.07, but still not quite at 0.05. So we focused at this point less on our treatment effect, which looks to be good, but what's going on with the sham group, because we have to get some kind of handle on that in order to reduce variability to show statistical significance. And so the first thing we looked at was something that was known to be present in the literature in terms of a spontaneous improvement that can be seen when you have a bilateral disease and you're treating the worst seeing eye. If that worst seeing eye is, if the disparity between the good seeing eye and the worst seeing eye is particularly large, then you can have just a spontaneous improvement, even if there is no treatment, when you're looking at the worst seeing eye. And of course, we have to dose the worst seeing eye for safety reasons as part of a clinical trial. So basically, everybody doing experimental therapy in RP is going to be faced with this kind of challenge, and similarly in AMD. And that is, if you have a big disparity between the 2 eyes, you have to treat the worst seeing eye, then you're liable to see some spontaneous improvement, and that can impact your sham control. And here's just an example of various studies reporting this in a variety of diseases. So if we go back to our clinical data and we take out everybody with a very large disparity, say a 15-letter disparity or worse between the two eyes going into the trial, so they haven't even been treated yet, and you just discard that data, what you see is that the sham effect still there to some extent, but it's much more subdued. The treatment effect gets a little bit of a boost. And now we have nice clinical significance at both 12 and 6 months. Now, we didn't just stop there, even though this looks like this change alone, adding this selection criteria would have given us statistical significance. We took the opportunity to look for additional things that might contribute to treatment effect, given our particular type of treatment and the type of patients we're looking at. And our reasoning was that in order to rescue cones, we're going to have cones there that are still left to rescue. So what are some of the ways that we can get an assessment of how many cones are still present in the patient's retina? Well, with adaptive optics, you could literally get in there and start counting cones, but that's kind of labor intensive. So are there quicker and easier ways of getting a handle on this? One way we decided to look at retrospectively was central foveal thickness. So with OCT, that's something that's pretty standard in ophthalmology nowadays. You can look at the central thickness of the retina right in the foveal center, and that thickness is composed entirely of cones. So you can get a readout crudely just with one single measurement in terms of cones right in the middle of the retina. And that appears to be promising, but we weren't able to apply that to our original data because we didn't have those measurements going into the trial. Another thing we looked at was central visual field. As the disease progresses, we know you start with tunnel vision and it gets smaller and smaller, and people start looking through a straw type of effect. At that point, you're really looking at central visual field. As that central vision decreases from the periphery in, people start to have more and more difficulty in terms of fixating on a target. And as this happens, their ability to perform visual tasks starts to degrade and become much more challenging. So if we used 8 degrees of visual field as a cutoff, and we could apply that again to our data, along with the original screening related to disparity between the eyes, voila, we get a sham readout that looks very much more like what we'd expect, at least beyond the first month. And so at that point, the sham looks like after 1 month, it looks to be just bobbling around the baseline, pretty much like the type of variability you expect from RP patients visit to visit. On the other hand, the treatment effect gets yet another boost, and we have significance from 3 months all the way out. You know, at both 6 months and at 12 months, we're exceeding a 5-letter difference, 15, sorry, which is a seemingly impossible task in RP. You know, this suggests that maybe it's not impossible. But anyway, the key here is just to get statistical significance. So I think we did derive a lot of very valuable data from this trial. And so going forward, we're getting through all these trials. And now we're just looking forward to the next trial where we're going to apply the lessons learned from this past one. And hopefully from there we can get approval. And I know a lot of people are eagerly awaiting that, not just me. So anyway, thank you very much. And there are other diseases we think that this will be applicable to as well. So thank you.
Really great talk, Henry. And I'm going to actually throw the first 2 or 3 questions that we've gotten to you, and a reminder to our participants. You can type questions in the Q&A section at the bottom of your screen. One thing I want to be clear on, when you were showing the difference between the sham and the treated eye, was that an improvement in vision for the treated eye as opposed, or were you comparing a treated eye to an untreated eye?
Well, that's very observant of you, and really kind of interesting finding, because going into this, I was thinking about neuroprotection in an old school kind of way, and that's why I made some kind of vague comments early in my talk that we might need to be open-minded about this. I was talking to myself as much as anyone, because we did see actual improvement. Now, that's not what you call neuroprotection in the strict definition, because how do you get vision back from just rescuing a neuron from dying? In theory, it just, the best you could hope for is maintaining your current status. So what I think is going on here is that you are rescuing neurons, in particular photoreceptors, but something else is going on. It's happening rapidly, and it's impacting the vision in a kind of profound way. And what I think is going on is that as the cells are slowly dying over years and decades, they lose the ability to elaborate the cone outer segment. So they retract those outer segments, so to speak. So it's like the cell pulls in the antenna, and the cell is still alive, but it's doing all it can to just kind of go into survival mode. And we know that these outer segments are hugely energy intensive. So they put massive demands on the metabolism of the cell. And if the cell's barely hanging on for dear life, it literally cannot afford to make an outer segment. But I think the signals coming from the retinal progenitor cells are basically like smelling salts. It says, hey, knock it off. This has all been a false alarm. You need to get back to work. And so they literally change their metabolic programming in kind of mid-death and say, oh, wait a minute, we're supposed to be working here. And then what I anticipate happens is that they regrow an outer segment. And when that happens in the macular cones in great enough numbers, you have this kind of amazing return of vision. Now, that doesn't mean it's normal. Even we've had some patients hit 20/20 again. That doesn't mean they're normal, but it's mind-boggling that this could even occur.
Well, fingers crossed moving forward. And I just want to make a comment. I think what you showed in terms of some of the challenges in designing a clinical trial, choosing the right patients, you get a sham effect, and that sham effect is not unique to the jCyte trial. You have to treat the worst seeing eye. This just reflects all the challenges one has when designing a clinical trial, and you kind of have to adjust things as you're going. So thanks for going through all that. The next couple questions I'd like to ask Kapil. So Kapil, your trial right now is for people with advanced dry AMD, which we call geographic atrophy, and you're transplanting RPE cells. And obviously those are the only patients you're going to enroll in that trial. What other diseases down the road might benefit from this kind of approach, or the RPE cell, or really the other cell transplant?
Yeah, Ben, excellent point. So the way it works with the FDA is that you get approval for one disease at a time for phase 1 study, especially. But the same RPE or similar RPE patch is being tested in London and has been tested in Japan for wet AMD. So we know that it has the potential to work for wet stage of AMD, which is also another late stage of AMD. Previously, a group had tested RPE replacement for Stargardt. So there's a potential to use it for Stargardt's. There's a condition called myopic macular degeneration. These are the patients that have childhood onset pathological myopia that in their young adulthood develop RPE degeneration, very similar to dry AMD. So it should work for those patients. Patients with Best disease, it may work for those patients. Patients with RP65 mutations leading to RP atrophy or RP4 receptor atrophy, it should work for those patients. So most of the conditions where RPE is degenerating or atrophying, it should work. But it's the way our system is set up, it's going to be one approval or at least one phase 1 at a time. Once we have demonstrated safety, then perhaps in the latest efficacy stages, we could combine and test in other diseases as well.
Right. Oh, well, that's exciting. I'm sure we have people affected by AMD, but we have people with Stargardt disease, other macular diseases. And while we don't at the foundation fund myopic degeneration research, we get a lot of questions on that. So it's exciting that that could potentially apply. Another good question we got was, how big of a patch can you design? Because you have the fovea, the very center of the retina, then you have the macula, which is still the center, but that extends further out. Can you cover the whole macula? Can you go beyond the macula?
Yeah, definitely you can cover the whole macula. So the macula is 5 millimeter in diameter. So the area wise, it would be right about 15, 16 square millimeter. So the patch we have is 8 square millimeter. So you could put 2 patches that would cover the entire macula. There are groups who are testing one single patch that is 16 to 18 square millimeter that would cover the entire macula. And as you would know that the macula is responsible for most of our central vision. So the goal is to try and preserve the central vision at this point. Can you go way beyond that? Potentially, yes, it's a surgical limitation. I think the goal in that case would be to try and develop multiple or deliver multiple patches in different parts of the eye.
Right. And I think an important point, and please correct me if I'm wrong, but the patient needs photoreceptors in that area to be alive so that the patch can keep those photoreceptors alive. If there's no cones there or rods, there's nothing left to support with those new RPE cells as well.
That is correct. For the RP patch, that is exactly right. But the future patches that we're trying to develop, which is a dual RP photoreceptor patch, there you would not need, there even with the areas where photoreceptors have degenerated, you should be able to deliver both of those combined, and they should be able to preserve vision in that part as well.
Right, right. Very, very exciting. Very exciting. So, Karl, thank you again for your great presentation on regeneration. I love to talk about it. I realize we're not there yet. This is a big question. What still needs to happen to get a regenerative approach in a human study? What are some of the next steps? And I realize it's not just one step. There's a ways to go.
Yeah, I guess this is a pretty technical question because there's many things, basic biology that we still need to understand regarding how to convert one cell into another cell in different contexts. Technically speaking, we talked about getting human cells to proliferate, right? That's one technical hurdle that people haven't done in the human systems yet, and really it hasn't really been proven in the mouse systems either. So this is something that will happen. I think there's been a lot of intensive research over the past year and a half, and I think that's a promising area. And then just steering them in the right direction. So after we're able to do these in little bits and pieces, I think developing a gene therapy that sort of jump starts the first process, gets the proliferation going, and then a second gene therapy vector, which pushes in the right direction. These are tools that the field is developing quickly. There's a lot of great AAV-based tools that a lot of labs have developed And so it's simply a matter of applying the basic biology know-how to those tools, and then establishing that they work in a human system. So, I think showing it in animals is one really good step, proving that you can cure some vision in a mouse. Also, as a preclinical model, putting it into an organoid and showing that you can kill the cells, the photoreceptors, and then bring them back. Once you've established that those things work, then I think it's a matter of just taking the safest AAV virus out there and applying that as well. So yeah, and I think there's a lot of companies in the in that space that are developing those kind of tools. So I think over the next year and a half, we're probably going to see a lot of activity in this area.
That's great. That's
Yeah.
great.
But things in the clinical world take a lot longer, and I'm admittedly a basic scientist. I'm not in the clinical space, but we do keep our eye on the ball. So
Right, right. And this may not be a fair question either, but I'll ask it. Is there a certain kind of retinal disease patient that you think will be most amenable to this approach? Is there a specific disease like RP or LCA? And should they be a person who's already lost a lot of photoreceptors, or can you treat earlier stage? Do you have a sense of that yet?
yeah, I guess that's a complicated question. I mean, you don't want to do anything that's going to speed up the course of disease. If the manipulation that you do makes things worse, well, then you're out of luck. Ideally, this is the kind of thing that would work after cells have been lost. So there's a lot of neuroprotective strategies. So Dr. Klassen was describing ways to delay cell death. And so if in the event that fails, well, as a fallback, we have another solution that we're working on, which would be to replace the cells that have died. And Kapil's work could potentially jumpstart that as well with a patch, which would, kill two birds with one stone there. A lot of times when rods die, cones die as well, RPE gets impacted. So maybe replacing a larger patch might be in order. In animals, that doesn't seem to be necessary. So I think, just jump starting the regenerative process might be sufficient for many of these things. But I think low-hanging fruit maybe is to LCA, RP. Of course, we're also working on retinal ganglion cell regeneration. So in the case of glaucoma, that would be the opposite there. So there are different strategies, but I don't think, some things like AMD, where you have a very large involvement of RPE cells, maybe, this approach isn't great for that at this time.
Right, right. And I think you made an important point that I hear a lot of researchers make. It's a lot easier to save what you have than to bring back what's been lost. Obviously, you three researchers are working on different approaches to do both, and we at the foundation are funding both approaches. But the more you can save what you have, that's just an easier hurdle. So Henry, back over to you. I have a couple questions. One question that came in. So your approach, you've shown efficacy out to 12 months of a dose of your intravitreally injected cells, and you mentioned that redosing will be needed. Do you have a sense, I guess the first question is, how often redosing will be appropriate for your patients if this moves forward? And secondly, have you tried redosing patients yet in a clinical trial?
Yeah, so first off, I don't know that the FDA would say we've shown efficacy, because a lot of people are really interested in seeing or the statistical significance, but I think it's safe to say there's a strong signal that I wouldn't bet against it, put it that way. So for all intents and purposes, it looks like there's something there. So redosing is really of interest, and the data we have so far shows that that signal that we do see persists quite well. And so the thing is, though, remember I showed that the cells form these little spheres in the vitreous. Those spheres don't integrate and they're not vascularized, and eventually they go away. So they don't last. They last much longer than a typical drug, but they don't last as long as, say a liver transplant, at least under ideal conditions. So you'd like it to be that you could treat somebody and that's it, one and done. But that not being the case, as the cells go away, because their role is secreting these factors, it stands to reason that the treatment effect will fade out. And that's our impression also from following the patients. Now, the data I have isn't as extensive in terms of like what exactly, we're looking at in terms of how quickly that effect fades out. So one of the things we've been looking to see is get some quantitation around the survival of the grafts. Well, that turns out to be harder than you might think, because all of ophthalmology is designed to either look at the front of the eye or at the retina in the back. And, the vitreous is just kind of in the way. So it's hard to get a real quantitative readout. Doesn't mean it would be impossible, but it's still a developmental kind of thing. Yeah, so in terms of like retreating patients, the FDA is very aware of the fact that this is likely to be a situation where we need to retreat. And that's why I've made a point of talking about the immunology of the situation, which we're further ahead on that than we are on some of the other aspects of it. Because immunology is either a big problem or it's not a big problem. It's really hard to be in between. And by big problem, I mean if you have graft rejection, because not only does the grafter get rejected, but there can be bystander damage. So a rejection event could be, a safety concern for the eye. But that's what we're not seeing. We're not seeing graft rejection events. And we're not seeing them after redosing. And yes, we have redosed, and we've redosed in two different ways. So after the very first trial, we had patients elect to whether they wanted to get the other eye dosed. Now that's the other eye, but from an immunological standpoint, it's a second kind of antigenic stimulus, if you will. So from the immune system standpoint, the other eye is virtually as challenging as putting it back in the same eye. But of course, putting it back in the same eye is also an issue because maybe the local inflammation and situation in that eye could provoke something that isn't elicited if you go to the other eye. So in our second trial, we had redosing in the same eye. And that appears to also be well tolerated. But there's still an issue of like, how soon do you redose? And, that's probably going to depend on how big the dose is. So as we're doing dose escalation, that's throwing another variable into this whole mix, because the more cells you put in, even if it's the same kind of bell curve, the longer they're going to last just because you started with more. So, that's all part of the equation here. But there's, I'm not recommending that we redose like a maniac and just inject as many cells as often as we can. I'd rather let them go away, get a nice quiet eye, and then start over again. But that's just my preference. But the more complicated you make it, the longer it's going to take to figure out what some of these parameters really look like.
Sure. Thank you for sharing that perspective. And my last question for you and Kapil, we have about 3 minutes until we'll go to our close here. Are you immunosuppressing at all? And I'll throw that over to Kapil first.
So since our trial is autologous, we are not immunosuppressing our patients. We do just local steroid challenge or steroid application during the phase of the surgery. So the eye heals from surgery, that's all.
Right. And that's huge. I mean, so many of these therapies you do need to immunosuppress, some to a greater degree, some to a lesser degree, but that's a real advantage of your approach.
And that was kind of the premise of us doing the autologous, because
Right.
especially in our case, the patients were older. We didn't want to give them systemic long-term immunosuppression, which raises a lot of other adverse effects on their body and their eyes as well.
Right. And Henry, are you immunosuppressing at all?
We're not, but we are using steroid drops after the injection,
Right.
similar to what Kapil was saying.
Right.
Because, to the extent we do see side effects, it's usually the type of thing that you associate with an injection of any nature into the eye. There's going to be some inflammation there, and the presence of cells might exacerbate that to some extent. But it appears to be generally well tolerated with a little bit of boost from some steroid drops. But we're using allogeneic cells, and so that's really pushing the envelope, because whereas if you use autologous like Kapil, the whole idea is that the graft would be seen as self, so would not be rejected. Whereas an allograft, unless you're an identical twin, there's the likelihood that some kind of disparity is noted at the MHC locuses, and the cells get booted out or killed on the spot is more like it. But in this case, they seem to be well tolerated. Now, this does not cross over into allogeneic transplants in general. So, the rate begs the question, why are these allogeneic cells being tolerated? And I think it's something I've taken a look at from various angles over the years, and I don't claim to have a comprehensive answer, but it's multiple factors, I think. For one thing, we know that the eye has certain types of local immune privilege, which means that that the tendency towards rejection is actively downregulated one way or another. And we know that from, say, corneal transplants in the front of the eye, where you don't typically immunosuppress people to give them a corneal transplant. That doesn't mean they can't reject, but the standard of care is to try to avoid using immune suppression.
Right.
And the similar situation obtains back in the retinal area and in the vitreous. But again, it's conditional. Now, the other thing is that these cells appear to be immune privileged. So they don't express class 2 antigen. And so they don't have a way to provoke the typical graft-mediated immunity. Now, I've also shown that they could express class 2, so they normally don't. But if you hit them with certain factors, you can upregulate it. So it's not that they're congenitally incapable of being rejected. It's just that they're in a state that does not lend itself to being rejected. In addition, they secrete a few things that seem to help downregulate the local microenvironment.
Right. Thank you, Henry. Thank you, gentlemen, for answering all those questions. The last portion of our program, we've learned a lot here today, a lot of diverse approaches, a lot of great information. Claire, Dr. Gelfman, my supervisor, please take us home with your esteemed perspective.
Well, thank you so much, Ben, and really a huge thank you to our presenters today. I myself have been taking copious notes. These are 3 really unique approaches to the treatment of inherited retinal diseases and dry AMD that we want to help support the treatment of in the very near term. It really speaks to the multi-pronged approach that we think about in terms of treating these blinding eye diseases. Frequently on these calls, we speak about gene-specific approaches, gene therapies, for example, gene editing. And for those, We talk a lot about genetic testing and the fact that at the foundation, you can get free genetic testing through the request from your ophthalmologist, and that's something offline. If anyone would like more information around, we're happy to provide that for you. But even with genetic testing, we don't always learn our causative gene, and even if we do, there may not be anybody working on a treatment specific for it. And that's why we get really excited about these approaches that really have a great potential of treating a degenerating photoreceptor to restore function so that our brain can interpret light that comes in that gets trapped by our rods and our cones. So we need to have a functional photoreceptor to see what's in front of us. And so I really feel like we have been privileged over the last hour or so to learn about these cell-based approaches. So whether or not we know the genetic cause, at the end of the day, we want to restore cellular function so we can see. And the three different presenters really capture the essence of three different ways of thinking about restoring cellular function. And, Dr. Bharti started with his human clinical trial that is in progress as we speak. And it's so unique because you keep hearing the word autologous and allogeneic. And his approach is autologous, meaning that the source of the therapy is the patient themselves. And so, this whole immune suppression, you don't have to worry about that. It's taking your own cells and giving them the potential to function properly, to essentially replace the lost cells with a functional one. And that approach, it's already in human clinical trials. We're going to pay very close attention to that because it really represents a unique way to think about treating. In the clinical trial, it's for dry AMD because it's those RPE cells that are being replaced. But as we discussed, there's potential to extend the learnings from that trial to photoreceptor cells, to a whole functional retina, so we can replace retinal cells as well. So we're going to look forward to hearing more about the progress of that trial. We also heard about the approach from Dr. Klassen through jCyte, which I think is also equally fascinating. And that's really more about giving the existing photoreceptors that are still there a chance to survive, to give them the food, if you will, that they don't have in order to survive in that moment. And we do that by providing cells, giving through an injection, and those cells aren't meant to replace the lost cells in the approach we heard from Dr. Bharti, but rather to secrete proteins that are needed by the existing cells to start functioning again. And so that approach is also, as we learned, in human clinical trials. And I'm excited, and I know, Dr. Klassen, we're all on the edge of our seats for that phase 3 clinical trial to expand your incredible learnings. I mean, I just want to focus too on the amazing amount of work that you and jCyte have done to learn from the data that came out of your phase 2 trial to then poise for success a future trial. And that is part of the clinical trial process, focusing on the word trial. We have learnings from every trial we do, so the next trial can be designed in such a way that really utilizes the learnings from the earlier trials to poise it for success. So I think that this approach of using cells to secrete proteins that are needed for the existing ones to prevent further degeneration, that neuroprotection approach, is one that we are all going to also be looking for and following very closely as that phase 3 trial gets underway. And so, the thing about cell therapy is it's really in its infancy. We're hearing it, we've heard about, 2 that are in progress in human clinical trials, but as Dr. Wahlin told us about, there's so much to learn, and why not take advantage of lower species, if you will, who have an ability to reprogram themselves. So you don't need to bring in something for the outside, an outside cell to either replace or provide sustenance for existing cells. And so I think a basic science approach through the work that's happening at UC San Diego in Dr. Wahlin's lab is going to allow us to learn about what cells need to be reprogrammed to begin functioning properly again. And I think that, it's such a unique approach from a basic science perspective. What are those factors that are needed? And then we'll take that information and then test those the hypotheses using the retinal organoid system that can be patient-derived specific or more of an allogeneic approach to learn about what factors are needed to endogenously to reprogram a cell to function properly. And so I really I feel like these are three very unique approaches that give us the ability to have more shots on goal, as we say. We spend a lot of time talking about gene-specific therapies. We talk a lot about mutation agnostic approaches that don't rely on knowing one's causative mutation. And what we've learned about today are these cellular-based therapies where we can, again, replace the cells that have otherwise died or atrophied or no longer there. We've also learned about an approach to reignite, if you will, cells that are existing but need to be kickstarted, if you will. And we've also learned about some work in progress to reprogram the cells that are still there rather than bringing in cells from the outside. So I think there's a lot of unique approaches for cell therapy to complement the other types of therapies that we fund and get excited about here at the Foundation Fighting Blindness. So I want to really thank all of our speakers today for three outstanding talks. Each of you presented your pathway very clearly. And I know I speak for everyone on the call here that we are very excited to see what's going to be happening in 6 months, a year, 2 years with respect to a cell-based therapy for the treatment of both wet macular degeneration as well as the different inherited retinal degenerations. So with that, I think we're right at time. Thank you again to everyone for joining this webinar. And we will, if we didn't answer your question live, then we will reach back out to you. Thank you again for your participation. And again, to all the speakers. Have a wonderful rest of the weekend and happy Father's Day to all.
Thank you.
Thank you.